Good morning, everybody. I'm Michael Brown, president of SAR. Most of you know that. And I'm absolutely delighted to introduce Christina Agapakis. Dr. Agapakis comes to us from Ginkgo Bioworks in Boston, where she's helping people to design microbes, right, that do useful things. And she's got a lot of interesting things to say. So the format is very simple. I'm going to do a handoff to Christina. She will simply set up a few issues that she wants to talk about, and then we'll just open up the conversation for maybe an hour and up to an hour and a half until we all get exhausted, and then we'll call it a day. But we're just delighted to have her here. Those of you who heard her speak last night know, I mean, she took us into a world that most of us don't know very much about, and that was really wonderful. And that's part of what our project is here in recent years. SAR has, as many of you know, a 111-year-old reputation in archaeology and Southwestern studies, which we still honor and is still part of our heritage, and we take it very seriously, but we're trying to push the envelope a little bit into areas that are less familiar to us and which we have reason to believe interest the general public. So you'll see some of our talks will be focused on Southwestern issues. We've got one on Chaco Canyon scheduled for January, if I'm not mistaken, on archaeoastronomy, and we've got a former student of mine, Liz Hoover, teaches at Brown University, who's talking about her. She's native, she's Mohawk, and she's going to talk about her comparative study of American Indian attempts to restore traditional diets and traditional agricultural systems. So those kind of themes are familiar to SAR members, but Christina's is new to many of us, and we've got one next month, again, a former MIT professor who works with new technologies, and she's going to talk about how they get used in education. So I give the floor to Christina Agapakis, and we'll just talk.
What should I talk about? Who saw? Did everybody go to the talk yesterday?
Yes.
No, there were some that aren't. Some that did too.
So maybe start all over again.
Tell your life story.
Well, I was born in Boston. No, I think I can talk about. I think maybe the best place to start is to start with some smelling and some more active things, and we can talk about some of the work that I do in biotechnology and at Ginkgo. So I'm trained as a biologist. I'm interested in what interesting things biology can do and what we can do with DNA. Ginkgo, as Michael said, we design microbes to do useful things. Some of our clients are flavor and fragrance companies, the companies that make the ingredients that end up in our perfumes and our foods. And they're looking for ways to source ingredients in ways that could be more sustainable, more accessible. So taking DNA from plants or other sources and putting them into yeast, those yeast can now grow and take that kind of information from the plant and make those smells, those tastes from the plants. It's sort of like brewing beer. So this project that I want to show for you guys today is using a very similar technology to try to make the smell of a flower that went extinct. We started with plants, a sample from a plant from the herbarium at Harvard. It was collected in 1912, and then very shortly after that was went extinct and was never seen again. And so from that sample, we were able to take a tiny bit of plant matter, break it down, extract the DNA, sequence that DNA. That DNA, we're able to, using information that we know about what makes plants smell, we're able to kind of identify some sequences that we predicted would be involved in making that smell.
Can I ask a dumb question about that? Because obviously, are you 100% certain that you got all the relevant sequences for these scents?
I'm 100% sure I did not get all of the
relevant
sequences. Yeah.
So when you get a scent, you think it's an approximation, but it may not be 100% on target.
Yeah. So all we could really find were enzymes. So sequences that code for single enzymes. Enzymes are sort of the machines inside cells that make chemistry, make stuff happen. Louder. Is everybody okay hearing me if I talk like this loud?
That's better.
And
okay.
a little slower.
Sorry, I am a fast talker. I'll try. Please interrupt. Obviously, this is supposed to be a conversation, so if there's anything I say that doesn't make any sense.
So you're actually not. It's not the scent, the code for the scent molecules. It's the code for the enzymes that produce the scent molecules.
Right.
Wow.
Right.
Okay.
And so there's many enzymes that are involved in scent molecules,
Sure.
and a flower could have hundreds of different molecules and hundreds of different enzymes. And so we're really looking at a pretty narrow range of things that we know for sure, and we have some biochemical expertise in and know a little bit about. So I'm already sort of, very sort of severely limiting what I can find with the experiment that we can do today. But that DNA sequence, though, codes for an enzyme. We were able to insert that DNA into yeast, grow that yeast, and then actually measure what is that yeast making that's different from what non-engineered yeast make. And it could come up with a list of molecules. I think for this flower we found 11 different molecules that are known to have some sort of scent properties. And I sent those 11 molecules to a perfumer. Her name is Sissel Tolas. I talked about her yesterday. She's a really interesting, she's trained as a chemist and a linguist, and she then trained herself for many, many years to be an expert in smell. She went all over the world just smelling everything, collecting smell, like just the most disgusting things you can imagine to like the most beautiful, and really trying to develop a library and a vocabulary for smell that she could then work with as an artist and as an educator to help people learn how to navigate the smellscape. And so she's got a really interesting perspective on scent and a really interesting approach to doing this kind of thing. And she was able to interpret that list of molecules in a number of different ways. So I just have 3 examples of what she came up with. Like I can just hand them out and we can start doing a little discussion, what you guys smell, what you notice about it. I can tell you first a little bit more about the plant. It was, last seen in 1912. It's a Hawaiian relative of hibiscus. It sort of grew on the lava fields in Hawaii, and it has this, like, the hibiscus flower, like in Hawaiian, like this. It's the hibiscadelphus one. It's like more tight, like the petals are kind of like this, so it makes a little nozzle thing. And then there's birds that would pollinate it, called the honey creepers. They have these really long noses, long beaks that would pollinate them, and that sort of ecosystem became disrupted as people were expanding into the lava fields, and it went extinct. So that's like where, situate yourself on the lava fields. In 1912, you see the last member of this whole species hanging on to the rocks, and imagine this.
So one more question. So you've got 3 different, slightly different scents, but are they all expressions of that same plant?
Yeah, they're all the same plant, but they're different. To kind of show you too that these are all sort of interpretations. There's always an interpretive act when it comes to kind of doing this process of reproduction.
Okay.
Yeah. So I have 3 strips with times 3 different molecules. So you take one and pass it around. This one is number 5, so you can take and pass it around.
Was there an attempt to actually bring the plant back?
No.
Is that just too difficult?
It would be too difficult.
Uh-huh.
I think so. So there are people who are trying to do things like bring mammoths back. Not kidding. But the way that they're approaching that is taking the elephant genome and then sort of swapping out genes with the mammoth genome that they have. There are hibiscadelphus relatives that are still sort of extant and sort of have survived. We haven't been able, we wouldn't really be able to do a full genome reconstruction from the samples that we have, and it's hard. It's hard.
The scent you can't.
The scent
the
you
actual
can't.
plant.
This one is number 10.
They moved at 5 on so
They all listen.
Sorry.
Another one's 10. Oh, so it's 3 different scents. She's got 3 strips of
They
the
have,
same
and
scent.
they all have numbers on them.
Exactly.
Which is a business.
Here, I'll trade you.
Anybody need a 10?
That's a 5. You want to see the
There's a
difference?
couple of
Not fancy, better than cheese.
Husband and wife have their mother. We're called the Notes. And one actually works as a secretary for and then all of a sudden they realized that she has Notes. Her husband, and it wasn't what you're talking about.
That's
It's
kind of
actually about training.
That's sex.
They just have this really
Weird.
that.
Hard for me to do already done.
It's trainable from what you're saying.
Oh, yeah.
It's also-
Anybody else think 5?
It's like somebody who has tons of testosterone that climbs mountains. That's
We'll have to find out.
Okay, we've got a 10 and a 2.
A 10 and a 2.
There's a 2 going.
Oh, now you got all 3.
The 208,
Just
yeah.
got two.
Which one is that?
These guys are hogging the
Okay, so I've done the 10.
This is
Because
Five.
everyone can train themselves a little bit. I think we assume that we have a bad sense of smell. We assume that we're not good smellers, that like dogs are much better than humans. Humans have actually pretty decent sense of smell. We don't think about it. We don't train it.
Okay.
So everybody can be trained.
Wait,
The
smells
other
like
side is gonna take a photo. Everyone can like do that.
Or do you have a two?
Here's
There are the
I
three.
haven't had two yet.
Here's
The
two
two
all
is
three.
a
It
little
talks about all
bit
three.
better.
Some people have a more
I've done the other two.
Right.
interest, capacity.
This
Laura
is-
said a
this is
cool
not-
thing about
Factors,
the
it's
kind
more
of
like
wine.
Yeah.
This kind of starts out the same, yet has a different
Actually, we did a
finish.
study where they had,
It's like the pot that what it leaves you with.
Actually,
is
Yeah,
that-
little chocolate on the carpet, and humans are actually.
The bitter one, number
Here,
two.
this is all three. So that's all, so try all three together. Okay,
Who
okay.
are the good smell?
So then you can see the differences.
It's like, I know, but I can't do that. So, but it's very interesting, this idea of the train.
The other side.
That one's more? Okay. I nominate Steve to tell us what the differences are.
No, but we need a three over here.
Oh, you didn't get the three?
Yeah, there's 2, 5, and 10.
What do you have? You have
Oh,
2 and
two
5?
then.
Okay, here's
Here's two.
Who has a 5?
Here's a 5.
Oh, no, you keep your 3 together. Your 3
Oh.
needs to be together.
We're missing the 10. We're missing the 10.
So let's, here, you have all 3. Tell us the differences. Oh, you haven't tried them at all. So pass them back when you're done. Okay.
We're lucky, you know, there are a lot of plants that do a dead meat mimicry.
Yeah.
Rotting meat mimicry. Thank you for not inflicting.
I did try to find cement-related genes in the mammoth genome.
Oh.
It turns out it's like the biochemistry is a little harder to make musks, and we didn't have as much expertise there.
How big is the genome of the plant?
The plant genomes are typically bigger than the human genome. They can be quite complex. We didn't do full genome sequencing because we just did a bait library project.
You had the woman that you discussed last night that was obviously an expert in terms of identifying different types of odors or aromas or whatever you want to call them. Was she able to take these smells and bridge them to something that is currently available in plant life that's alive and still prosperous and available?
I
That
think
it's sort of like bridging something from the past to something that is maybe it evolved from?
I don't think that's the approach that she took, but we have worked with other perfumers and with projects like this. Sort of before we were able to do a lot of the sequencing work ourselves, there's another flower that has been, somebody had done a little bit of sequencing and published some information about it, and based on that we made some guesses of what we would have predicted it would have smelled like, and they were able to also find biologically like what other plants would be related, and there are like well-known perfume plants that are, flowers that are used in perfumery that were related to that flower, a separate project, and we did work with a perfumer who was able to kind of, create that sort of the extant flower, but like a cousin, and kind of used the known perfumery scent of that flower and kind of like echoed it into the name for the extinct project. So it depends on
Are the perfumers interested in these scents? I mean, I know that some of the classic things like sandalwood are actually becoming scarce.
Yeah.
So they're
Sandalwood is a
looking
great
for
example.
alternatives.
Yeah. So there's synthetic like chemistry variants of a lot of sandalwoods, and people are trying. It doesn't give you a lot of the depth, and some of the molecules are hard to create. Sandalwood, there are definitely projects not active at Ginkgo right now, but within the community of folks trying this approach to produce those molecules, so you don't have to cut down trees to get to the
Wow.
scent. Yeah.
Interesting. So then how does that, from the business side, we talked about this a little bit last night, but I mean, what is Ginkgo selling to? Are you licensing? How are you working the business part of it?
Yeah, our approach is to license the organisms to our customers.
Can you talk louder?
Sorry. Our approach has been to license the organisms to our partners. So in flavor and fragrance, our customers are not actually experts in biotechnology and bioengineering. So typically we work pretty closely with them as they manufacture the products. They'll take the organism, we'll put them in a big tank together, we'll be able to extract the molecule, and they'll sell the product, and we get royalties on that product that they'll be selling. But other customers we have are, for example, Ajinomoto. It's a Japanese company that makes food ingredients. You might have seen it. It means like tasty or something. Ajinomoto, they make a lot of different food ingredients, and
It's like MSG. Right?
MSG is one of their products, but we don't work with them on
It's
MSG.
huge in Latin.
Yeah. MSG is great. It's been wrongly vilified. We can talk about that. But we work with them, and they're experts in fermentation. They historically have done tons of fermentation to produce these ingredients. And so for them, the relationship is much more. We can really hand them the organism. We can license it to them. They can put it in their tanks.
Are they looking for the ultimate umami flavor, or is that one of the?
That's not what we're working on with them. We're helping them use their resources more efficiently, so that the strains that they work with to produce their products are able to be more effective, and so they have less resources.
When you say that you're trying to make that more efficient for them, does that mean that you're really trying to isolate the genes for them? Is that how you?
There's a lot of different approaches
Yeah.
that you could take to make a strain that's already doing fermentation like this more efficient. So you could have ways of using the sugar more efficiently. So maybe you delete something that is consuming sugar but not going to the product, or you could have it teach the cell to eat a different kind of sugar that is a cheaper or more renewable source of a feedstock for the cell. Or you could optimize the genes that are doing the conversions, the enzymes inside of the cell that are actually making the final product. You could optimize those and make them more efficient. There's lots of different approaches that you might take.
But it's all at the genetic level
Yeah.
that you're talking about. Right?
Yeah.
So you say that yeast is doing a lot of the heavy lifting for you.
Mm-hmm.
So how does that work? So let's say you got a yeast and it's producing one of these scents. How do you collect it, or am I missing? There's some step I'm missing here. Like
How do you get the scent from the yeast?
yeah.
So sometimes they'll actually secrete it, so it'll kind of be in the beer broth that the yeast is making. And then there's chemical techniques that you might use to separate it out. If it's still inside of the cell, you'd have to first kind of pop open the cells and then do the chemical extraction. But yeah, it sort of becomes part of the broth of what the yeast is doing, and then there's a lot of chemistry that happens after the biology to make the product.
Can you talk about why yeast? Because yeast is like the thing that I feel like biologists are like, yeast does everything, but like why yeast?
Yeast is awesome, right? Yeast is it's a historical thing, right? So like people have been using yeast to brew stuff for thousands and thousands of years before we knew what yeast was. So you can make beer. We're really good at doing that. We're really good at growing lots and lots of yeast. And as a result, people have also used yeast as a model for understanding genetics too, because it's around. Like fermentation was some of the first places, the first applications of people learning what microbes were, learning what yeast was, and then learning deeper and deeper what the genetic and biochemical things happening inside cells are. So yeah, there's sort of a lot of historical reasons why yeast becomes sort of the.
But mechanically, why is yeast a better thing to use to insert this DNA into rather than some other?
So E. coli is another one that people use a lot of. So we have products where we're engineering E. coli to do that kind of manufacturing. Yeast just has that sort of the fact of brewing and the fact of the resources that we have to grow at yeast at really large scales makes it a little bit easier sometimes, but there's other reasons too that you would prefer to use E. coli. You'd have to retool your fermentation tanks to work better for E. coli, but the concept is the same. So we don't always work with yeast. We work with a number of different organisms, so different species of yeast, not just brewer's yeast, which I just learned from Twitter. So somebody said this was I can't believe I made it to my mid-30s before I realized that cerveza in Spanish is the same root as Saccharomyces cerevisiae, which is the Latin name for brewer's yeast. So Latin cerevisiae and cerveza are the same. And I was I also made it to my mid-30s without knowing that. I'm a biologist. So that was interesting. Yeah. So I think
Thank you, Twitter.
Thanks, Twitter. I like that. I learned a lot. So,
My knowledge of these things is extremely limited. Soy is also fermented into like soy sauce.
Right.
Is that a parallel kind of process to what you use yeast for?
yes, it's a great question. So I don't know the species they use to ferment soy for soy sauce, but it's another microbe. It's probably another fungi that's probably related to yeast, but it wouldn't be the same as the brewer's yeast.
Which is why it has that umami thing, because it's in the mushroom end
Yeah.
of the spectrum.
And so there, as you know, so fermentation is just a kind of like, it's a chemical transformation that happens inside of cells. So it's the conversion of sugar into things like alcohol if you're making beer, or acids if you're making things like vinegar, sauerkraut, soy sauce. All those acids. That's the end result of the fermentation process. So fermentation is just the general term for those kinds of biochemical conversions that are happening. We ferment in our bodies. So if you don't have a lot of oxygen, you're exercising, you have a cramp. That's the acid that your cells are making because you're fermenting instead of doing normal biochemical respiration. Because you're kind of using fermentation to kind of get energy.
Instead of rinsing with drinking enough water, you're fermenting. Okay.
That's why I drink the juice.
There's also some crazy things where if you have too much yeast in your gut microbiome, you can actually ferment and get drunk from your gut because it's making alcohol like in your body. It's very rare, but there's some reported cases.
Talking to the officer about that.
Yeah.
Officer,
It's my yeast. I promise.
Actually, there's a famous Perry Mason case that I remember from when I was 10 about some guy who was arrested for driving while intoxicated, and Perry Mason brilliantly shows that for some medical reason he was producing his own alcohol and that he was innocent of an intentional crime of driving while intoxicated. He couldn't help himself. And then so in the courtroom, they give him crackers or something, and he proceeds to get completely drunk after. See, it made a real impression on my 10-year-old mind.
crackers.
I tried it, didn't
Had the beers that you've now.
Excuse me, is brewer's yeast the same as the kind of yeast that you use in bread?
It's very similar. I think it is the same species. Sometimes there'll be different kind of subspecies that are kind of specialized for different things, but they're all the same.
Okay, thank you.
I've read a number of articles on the 7 different ways that our eyes convert electromagnetic energy into frequency, intensity, send the way we see. I haven't read any articles that says how do the nerves in our nose convert molecules, of which there are a bazillion different types, into electrical signals that we differentiate. Are there any articles out there that says how we really smell things? I haven't read any.
There's tons, and they're super interesting, and there's actually like a really interesting like scientific controversy about how we smell at that like atomic level, which you might find interesting.
Yeah, down at the, because here's a fairly complex molecule.
Yeah.
And I've got to take that information and convert it into an electrical signal the brain can use.
So there's a class of receptors that lives on the membrane of a lot of different cell types and in your nose, the sort of olfactory cells. They're called the G protein coupled receptor, GPCR. GPCRs there's many different kinds of them. Each genome, like human genome probably has like 300. I think the mouse genome has like 1,000. Dogs have a lot. Each of those encoded receptors that are made of a protein that lives in the membrane of your cell in the olfactory bulb will interact with a molecule or a set of molecules that exist. So I think they've found that there's trillions of different molecular combinations that your receptors can detect. And the G protein coupled part is how the signal gets transduced from the receptor down to something that actually triggers a neuron to send a message to your brain. And that's just sort of a biochemical cascade. And different organisms will use different ways of doing it, but yeah, in humans it's sort of biochemical transformation.
So is it sort of like a receptor is tuned to a particular molecule?
Yeah. And so the controversy, which is very interesting, and I don't actually know where I stand exactly. I think I'm leaning towards the more mainstream interpretation, but I appreciate that there is a heterodox interpretation. Is that what the receptor is detecting is not the shape of the molecule, but actually like the quantum vibration of the molecule. So that that you have a quantum mechanically tuned detector in each of your olfactory receptors. And so there's been some interesting experiments that kind of prove and disprove this theory, and a lot of there's a very brash scientist who's the one who's kind of proposing the olfactory vibration thing. His name is Luca Turin, and he wrote a book called The Secret of Scent. And then there's a book about him called The Emperor of Scent, which is pretty interesting. Just because he's a really interesting character. He's a perfume reviewer. He writes like a big review book, and he's got this kind of out there idea about quantum olfaction.
You got to deal with it down at the really, really low level.
Yeah. And there's some there's some things that are confusing about those smell receptors where molecules that are almost identical in shape, but have you've probably seen like an image of an organic molecule with like hexagons and little lines. So you can imagine like two that look almost the same, but one has an oxygen here and one has a sulfur here. Shape is very similar. Quantum vibration is different. They smell very, very, very different. And then there's other molecules that have very, very different shapes that smell very similar to each other. And so like those inconsistencies make it very hard for people to map receptors to smells. And so and kind of opens up space for these different kinds of theories, which has been sort of interesting to track. I like to see that scientific controversy play out in just the past few years.
I understood that a lot of scent is a very strong memory inducer. So, but it seems to me from what you're discussing that if your perception is off the day you first smell it, because you had a cold that day or whatever, are people misperceiving smells when they smell it 20 years later unexpectedly if they haven't smelled it in between? Or
I don't know. That's an interesting question. So this Luca Turin guy in the Secret of Scent book, he's like, he's all these psychologists. They think that smell is unique for memory. It's all bullshit. He's like, the reason why is because we have such specificity in our smell that we don't necessarily have in other senses. That you can have you have a lot of precision. If you smell the same thing again, there's only one thing that would be that, where everything is a little bit different. And so that's what he says is can help trigger memory. Other people say things like, the nerves of the olfactory bulb go straight into the sort of memory center, and that there is some sort of primordial connection there. I think, I don't know. I don't know what, it's also hard to put words to it sometimes. You can have that flash of something, but you can't quite remember what it is. And I think that's just memory is complicated, smell is complicated.
I don't know if it would complicate the research into people, if you're trying to research people's ability to identify or differentiate, how much of that is getting, instead of just being a chemical neurotransmitter pathway back and forth, but it's getting wrapped around memories and other perceptions of what it means in their ability to just respond accurately, not intentionally or unintentionally.
That's a really good point. So the emotional part of smell and that memory and the association that we make with it absolutely affects our interpretation of it. So the Sissel's work in particular is all about this. So she says nothing stinks but thinking makes it so. Because we always have memory associations, we have cultural associations with different kinds of smells. So I think a really good example, poop, most of you would say it smells bad. I talked to a woman who grew up in rural India, and she's like, poop smells like clean. Manure, I use manure to clean my floor when I was a kid. And when I smell cow manure, I think my house is clean. So if the association that you have with something is really powerful. And that memory that you connect to it, and that's what drives a lot of our perception of what that smell is. So another example of it, which I think is really interesting too, there's a study where they called it olfactory hallucination, where they give people 2 bottles with identical, like they give you 2 strips. They're completely identical, like I dipped them in the same bottle, but I labeled one cheese and I labeled one vomit. Completely identical, people have a very, very different response and perception of that same molecule because of what the word is associated with.
Dolly, you mentioned it.
I wasn't there last night. Would you take a moment to go back? Can you back up just a minute?
Sure.
All right, so I've come to your building, I've come to your research facility. I walk in, all right, so you are doing research on different chemical molecules, right?
Yes.
And I'm assuming there are vats all over the place, right? All right, so how do you...
I wish I could show you some pictures. I got my computer.
I wish you would. So, all right, so then I'm a customer and I need some of whatever you're making available to me. How do you harvest it, let's use that word, and get it out the door to me?
So okay. Our lab, if you come in, if you're in Boston, let me know. I'll give you a tour. You come in. It's in an old army warehouse building. So it's a huge open space, sort of columns everywhere, and then we have glassed off in the center about like 6,000 square feet of lab space, which is full of robots. And people interacting with the robots. Everything is very small and automated. So it doesn't look like a typical lab. It also doesn't look like the brewery tanks. Those happen outside of Ginkgo's lab. So a customer comes to us, we show them around, we show them what we can do. Because what we can do is engineer yeast and the DNA of the yeast to make things and to measure what those things that it's making. And to tell you very precisely how much it's making and to make it sort of do it to spec. So we would talk with our customer. Our customer might be a Ajinomoto. They say, we want to be able to make this product but use 50% less sugar. And we say, okay, let's talk about how we will do it. And we would sort of think and study and figure out the DNA that we would have to change. Then we have in our lab, part of our lab is DNA synthesis, machines that actually chemically synthesize DNA strands and genes. And we can make those genes, and then we have other machines that will put that DNA into the yeast. And we have other machines that help us grow the yeast, and then other machines that will help us look at what is going on inside of the yeast cell. How have we changed it? How is the chemistry now different? And then we would send the final strain to the big brewery tank, and then we will be able to extract a thing that's like, okay, here's your product. And our customers are the people who would sell those products. Does that make any sense?
Yes.
Okay.
I have a quick question with respect to the individual human being with respect to smell.
Yeah.
My background's marketing, as I explained to you last night, so I'm more concerned about how people respond to the various tastes and smells. Working with products on a global basis, found that in different parts of the world, people responded differently to smell and taste. And the question I have as to whether or not that was a biological reason for that, or was it more of a mental, or what I call experiential, cultural issue, because it sort of suggested early on when we were talking about what was going on in your sensory perception.
I would absolutely put it on cultural rather than biological.
Because it would be, so in essence, someone if you transported from one part of the world to the United States, for example, that in a period of time they would completely acculturate in terms of taste as long as they were experiencing it here?
Yes.
Okay.
So for example, like my parents immigrated from Greece, they grew up with different flavors and tastes and things that they liked, and than I did as a kid growing up in the US. We have the same DNA, but I prefer different things because I grew up in the US versus Greece. And similarly, I think you see too, like culturally, food practices and habits are shifting. Like there's more westernized, more American diets being exported, and people are changing their taste over time too. And vice versa. Like I think U.S. tastes are also like becoming different too. I think there is a more diverse kind of palate, and like there's more I think experimentation when it comes to flavors. At least a certain class of American foodies.
I'm sorry. How much does sight, the actual experience of looking at food, as long as we're in
A
food,
lot.
doesn't that have a great deal to do with all this?
Totally. Yeah. Yeah. There's a lot of really interesting studies about like senses being sort of influencing each other. So like even what you hear while you're eating. So like yeah, I mean if you worked in the food industry and marketing, like the crunch of the food, the crinkle of the bag. All of those things are carefully calibrated and studied because those are all influence how we experience the taste of the thing.
This is just an anecdotal cultural comment. A couple of decades ago, Rena Swenzel, who was an anthropologist from Santa Clara, her family, she was telling me that as children they ran through the Pueblo and they would taste the houses. And if you think about from a chemistry point of view, I think in terms of ceramics, clay, you would go, oh well, you know, they're getting their dirt and filtering it for the pottery from the same place, but they actually distinguish between what the houses taste like. And I was like, whoa, I never would have thought of tasting a house. But it's all that, it's the dirt. You know? Anyway, I just wanted to throw that out, that it was right in our backyard here. So.
In the part of the Amazon where I worked, it's pretty common in the Western Amazon, the staff of life is a beer made from manioc or cassava. And the way it's processed, this is sweet manioc, so it doesn't have to have the toxins extracted. They they peel it, slice it, cook it, mash it up, and then as they're mashing it up, they chew it and spit it back into the bowl to convert the starches into sugar. And adults drink gallons of this stuff a day. I mean, the first thing that a woman does in a traditional household about 4 a.m., she warms up a bowl of masato or nihamanch, this is called one of the people I work with, and takes it to her husband. And I became a connoisseur of this stuff because I didn't have any options really. And it was striking to me that some women, their beer it tasted really good, and then others was kind of off, and I had no idea whether it was the microbes in their mouth, or whether it was something in the ceramics that they fermented it in, but there were some women's beer that I just liked a lot better, and whenever there was a party at their house, I was the first in line.
That's so interesting.
And for a lot of people, it's really disgusting, the whole idea of drinking that sort of thing, but it's amazing what you can adapt to when you don't have any, a choice.
I reviewed a paper where somebody was sequencing the fungi and the bacteria that were present.
So what did they find
beer
out?
in Ecuador. I don't remember. There was a pretty good. They were trying to correlate the different mouth bacteria with the different to show that it was there. I think it was actually difficult to prove that what was directly in the mouth was in the beer, but there was some correlation.
Sure. I mean,
they needed more experiments, but it was a good paper.
There would be residual, bacteria and yeast in the fermentation ceramics and so on. So it would be tough to sort it out. And sometimes it was very acetic. So if they let it get hard, it really was acetic. But on an everyday basis, it was only slightly alcoholic. And they serve it to babies too. It was really an alternative to mother's milk, and they learn to drink it very young, but with microscopic alcohol content. Can we shift to the safety issue that you raised last time? I'm sure people have questions about just how worried should we be about monkeying around with the bacteria and these other life forms? Because obviously there are risks of contamination risk. There's obviously the more sinister risk of biohazard, biowarfare. How do you described the lab, it didn't sound like you went through some kind of containment. structure or anything like that. So how do you think about that, and how does it play out in your work?
So in our lab right now, it's so I can give you a little more context, I guess, about how biologists look at biosafety, because I think that's a good frame of reference for thinking about the other pieces. So there's sort of legally and like federally regulated 4 levels of biosafety. So 1, 2, 3, and 4, as you go up, it gets more and more dangerous. Biosafety level 1 is perfectly safe. Like you could go in the lab and lick things. Don't, because there's probably like chemicals and stuff that are not as good. But from a biohazard point of view, it's fine. Like you could drink. I have partaken of the beers that we made. Like those are made with BL1 organisms. So yeasts, things that have a long history of use, things that are non-pathogenic and known to be because of long experience, are classified as BL1 organisms. And as long as you're not introducing DNA from organisms that are not BL1, you stay within the BL1 regime, which is like you don't need to have any sort of special safety equipment, only what you like need for the chemicals that you're going to have around you. All you need to make sure is that there's a locked door, so you can kind of like keep your stuff like isolated as biosafety level 1. So all of our labs today in the main facility for Ginkgo are biosafety level 1. And so yeah, all of you are welcome to come visit. We have goggles available because there are chemicals if you want, if you're worried about it, but it is safe. People work with it every day. It's safe for everybody. Biosafety level 2 is where you start getting like a little bit more concern. Those are usually things where people who have any immune deficiency might be at risk. So sometimes organisms, microbes that are present in the soil, microbes that are coming from the human body, many sort of like potentially pathogenic organisms like a staph, those would be biosafety level 2. So there you want to make sure you're wearing gloves, you're wearing a lab coat. Those have a little bit more restrictive policies. We do have a biosafety level 2 lab down at a different level. We don't let guests and visitors in. That's for we study soil bacteria in that lab. To give you more context, like I think HIV and research on HIV and those sort of human pathogens like that is BL2 plus. So there's a little bit more regulation, more restriction and safety processes. Level 3 is like you need a respirator. That's like you're studying tuberculosis, you're studying things that are airborne pathogens that are known to be dangerous. So flu, a lot of the kinds of things that you'd be studying in about kind of pathogen infectious diseases way. It's biosafety level 3. I've never worked in a biosafety level 3 lab. I've never actually even seen one. That's rare. And then biosafety level 4 is like Ebola, hot zone, the full thing. Like there's very few labs in the world that have that level of classification. So that's just like context to give you a sense. Like so when the kinds of engineering that we're doing, if you're taking genes from like well-known food plants and like things that are and moving them into well-known food safe microorganisms, there's very little concern for that, and there's things that you would check and monitor for, make sure you're not introducing new kinds of chemicals in what the yeast is producing, but overall like is pretty safe, and I'm very confident about the safety of those kinds of things.
But what if you produce, let's say, a yeast? We didn't talk about pharmaceuticals, but let's say you produced a yeast that could generate some pharmaceutical, and if that escapes into the wild, do you have beer that's producing, something that you don't want people to be?
That's a really good question. So, by and large, what the organisms that are engineered to produce high levels of any compound are not really viable outside of the lab. They can't compete with the wild organisms that are not doing this kind of thing.
Are they engineered to be less resilient than the wild varieties?
No, it depends. It depends on the context. They don't have to be because if you're convincing a cell, encouraging a cell to spend a lot of its energy making something that's not useful for it, that you're going to actually take away and give to somebody else, it's not going to be very happy with you. It's not going to be able to grow very fast. It's not going to be able to compete against a cell that's in the wild in a wild ecosystem. It's not going to grow. So there's again like decades of safe examples of safe use here, where there are like strict containment practices. There are ways that people are kind of monitoring drains and disposal of cells and all of those things, especially in pharmaceutical contexts. But yeah, those systems are kind of making sure to keep it in. And if there have been things that are released, I think it has not caused a problem because of that.
But then we know that microbes of different species can exchange, genetic material. So
Sure.
you don't worry about that, about these.
The kudzu of the microbe world. That's,
So again, like it depends on what it is. So I think I would worry about things like antibiotic resistance moving around. So if there were a strain that's going to go into production, you don't use the antibiotic resistance as part of that strain. Typically, we use antibiotic resistance quite a bit within the lab as a way to kind of keep track of and move pieces of DNA around, because you can very easily make sure that all of the strains that you have in your test tube are the ones that have your DNA, because if you add the antibiotic, it'll kill off all the other ones. But you don't want that antibiotic resistance to be like out in a bigger context. So by the time you get to production, you remove that DNA. Otherwise, yeah, if the genes are, depends on what they do, right? Those enzymes are all, where they're from, what they're doing. They're not going to necessarily produce a different. Yeah, you're not going to turn your yeast into Ebola, right? Like it would be like waking up one day and you're a lizard, right? Like that, and not to say that not to diminish the concern at all. Like just to say that the kinds of I think you have to be specific about what you're worried about moving around and what that kudzu could be.
right, it could be, I mean, to my knowledge, kudzu isn't inherently evil, it just has been wildly successful to the exclusion of some of the things that were there before it,
So I've never seen a lab strain that's more successful than the wild strains. And
What do you do with what you're not using? I hate to use the word waste, but all right, so you have manipulated something into something you can use. There's got to be
leftover cells.
Leftover stuff.
Yeah.
What do you do with it?
It depends. It depends on what it is. So in a lot of breweries and a lot of kind of fermentation things, infrastructure, breweries, and things like that, you'll have spent grain, and you'll have like yeast mass at the end, like biomass. For normal brewing, a lot of that yeast will end up going into kind of agricultural uses, so like as fertilizers or as part of animal feeds. So people will reuse a lot of that. People are also looking at ways of reusing spent grain and yeast biomass as a source of energy for another fermentation, to make something else that's useful out of it, to get the most out of their products. If you have something that you're making that is like a chemical that you wouldn't want to put in an agricultural context, that becomes more something that you would have to dispose of as depending on the hazard, what it was. And I don't know specifically like how pharmaceutical manufacturers would do that. It probably depends pretty highly on what it is.
But what do you do?
What do we do? So like within our lab, we have pretty small volumes of everything. So most of the time, it's disposed as waste. So we would bleach the cells to kill any of the live organisms, and then it would be disposed of as biohazard liquid waste. And depending on the chemical, it has a different safety regime.
You mentioned that your work with soil microbiomes. I assume that has agricultural implications. What exactly are you doing with those?
So we have a new joint venture. It's about a year old now with Bayer. Bayer is a big ag company. That company is called Join Bio. This is a new sort of joint venture spin-out company from Ginkgo. They are working on ag biologics. So microbes that you would sell and put either on seeds or spray in agricultural context that could help promote the growth of plants. So one of the projects there is bacteria that can fix nitrogen from the air and turn it into nitrogen fertilizers. So that's a kind of holy grail for ag. I think if you can make bacteria that can provide nitrogen in the roots as it's needed in the plants and not use artificial chemical additives in agriculture and fertilizers, that's a huge gain when it comes to sustainability and greenhouse gases. So like that's one of the kind of big projects, but there's a lot of ways that you could think of how bacteria might also be useful. Use bacteria that can kind of diminish the growth of fungi, so you don't have to add extra pesticides that could be that are used to now like limit the growth of some fungi or nematodes or whatever it is that you're worried about. So kind of more like biocontrol kind of context. That's one of the approaches. But those are all still research, so pretty early on. There's no products yet, and there would be quite obviously a lot of safety questions and tests that would need to happen before they get out into the field trials.
You mentioned last night in your talk, on the front of your computer, you had all these little decals.
Yeah.
And one that you pointed out was, I heart GMO.
That's right.
And I think a lot of us in the audience said, that's interesting. But could you just expound on that, about your thoughts about GMOs?
Totally. So I've been talking a lot about GMOs, genetically modified organisms, the yeasts, the things that we work with. I think genetic engineering is really interesting. It's really powerful. I think it has a lot of potential to do important and good things. I think GMOs have become, like the word GMO has become this villain, this boogeyman. It's just associated, like GMO equals bad. To the point where even within the industry, like within people who buy technology, people are saying like, oh no, like it's not GMO, it's CRISPR, it's something else, it's something different. I think that's silly. I think it like we should talk about what like it's genetically modified. This is why we're doing this genetic modification. I think it's interesting. So I do want you to say, that's interesting. Why would you say you love GMO? Doesn't mean I love Monsanto. They're not my favorite company. I think they do things that are wrong. I would disagree with. But I don't want to throw the baby out of the bathwater, I guess, is what the GMO thing. Because I think there's a potential, right? If you can decrease the use of artificial fertilizers using GMO technology, that's a big one.
Well, isn't an inoculation a GMO? I mean, if you get a flu shot, isn't that genetically modified?
I don't know how they do the flu shots. It depends, but sometimes yes. There's a lot of genetic engineering in a lot of products already, in a lot of medicines. I think GMO has kind of come to mean things that Monsanto makes in corn. But again, genetic engineering is so many different things. A lot of not vaccines. I don't know about vaccines. Some probably do have a genetically engineered component. Certainly insulin. The majority, like I think half of new drugs approved last year were biologics, were things like antibodies. Or protein-based therapies that are made in genetically modified, more modified cells. There's a lot of things that are genetically modified, and there are a lot of potential for really great things that we could do. And I want to call attention to those things and kind of talk about and even ask the question like, what would it take to love a GMO, right? Like, what are things ways that we could make?
Well, at this point in time, are you really looking at changing the terminology rather than correcting the impression of GMO? Because
that's a good question from a marketer. What do you think?
No, it is, because I in the past had to do it.
Yeah.
When I was at Colgate, we had an issue with respect to an ingredient that we had in a toothpaste, and it was one that we advertised. And someone came up with a claim that one of the chemicals that we were using in the toothpaste would create cancer. When we got into it, found out that someone had to consume a 12-ounce tube of toothpaste daily to even have the potentiality of it. But rather than fight it, we literally modified the formulation, added another ingredient, and then changed the entire selling story. Because it wasn't worth the fight to take on the antis who had come up with this, because there's some professor who used it for their doctorate theses. So the real question with respect to GMO, because that seems to be, the bad word in the world today in terms of what the work you're into, is the story then to change it and put it in a more positive spin as to what CRISPR is or something else that becomes more consumer friendly, because most of us have absolutely no clue what you're doing. All we know is what the outcome is and what ends up in the front page of the New York Times. So I think the real question is, what is the industry doing to protect science because of all the positive aspects of it rather than all the negatives that seem to be drummed up?
Do you have a comment on that, sir?
What can I add?
Yeah.
So I worked for a company for a while that is probably one of the largest GMO companies in the world. It's Chinese. And two things. One is this anti-GMO thing is a Western thing. Okay, it is the United States and Western Europe, first of all. So to your question is how to resolve it. It's a very local thing. Okay. And the second thing, which is a dilemma, and you were talking about it a bit last night, is GMO is gone. The train has left the station. The world is going GMO. If you're going to feed 9 billion people, it's going to be because of GMO. Okay? And so I think that the part of the conversation last night of how, going back to what you said, how do you break, the bad mistake that environmentalists did 25 years ago about GMO being the big evil thing, and whether that's through straight education, which is more science-based, which is, I think, the question last night of, okay, I used to, do animal husbandry and breed crops, and now you're doing, what's the difference? So that's kind of a basic thing. And the other thing is using other things like art or scent or other things to get people to kind of raise their level of understanding.
But I want to take your statement and take it. Okay, so let's assume that you got the Chinese who say, we look at this as a positive.
Yeah.
So therefore, people are more accepting of the research and the science itself, so that they fund research in that environment, they push it in the universities and what have you. Now you come to the United States, and all of a sudden, this particular science, I'm going to call it GMO, for lack of a better word, has such a bad connotation that everybody shuns it. So it puts a nation at a disadvantage. So when you start talking about pursuing some really positive issues,
Yeah, I read a paper just 2 days ago.
That's the thing that and I look at when I see some of these things, the terminology is such that it shuts doors that shouldn't be shut. We did that, saccharin was a case in point, because I was working at Nabisco and went through that whole thing with saccharin, aspartame, and all that, and found out that saccharin was safer than the artificial sugars that we use today. But you can't go back and make saccharin good. It's dead. and mainly, and I've seen that repeatedly. I mean, it's not just one. I've had a couple of experiences that way. That it's a shame because it shuts down certain areas of studies out of just sheer ignorance, or somebody deciding, I can make hay by shutting down this industry because it'll be helping mine.
But it's also a local. One of my points, a local shutdown.
Understandably, but it's still,
the
like
world,
you said.
the world's already.
No, I understand that. I think it puts us at competitive disadvantages in some cases.
But
and that's one thing.
Science you may share a different view on it, but science kind of doesn't care what the public thinks about GMO. Science moves forward.
It depends.
And the
So
public
it depends
has
on the
to-
context, right? So I think like pharma, like everything's GMO, that's the way it's going to be forever. It would be a huge loss to human health if we stopped using GMO to make medicines. It would be crazy. I think when you're talking about food, where that flashpoint exists, it's marketing's fault because I think it's oh, customers don't like it. Okay, non-GMO. I'm gonna cut it out. I'm gonna take that approach rather than something that is more active. So we've had now everywhere you turn, every store, every package says non-GMO. Non-GMO is a good thing. It's very hard to turn that around. It's true, but I can't also stop using GMO because any other word, people are not stupid. They're like, this is genetic engineering. This is a GMO. You're lying to me. That's worse. If people think that you're I don't know. I do want to talk about it as what it is because I think that's a word that people know. That's a word that people fear because there's been marketing to say that non-GMO is good. So I don't know. Yeah, my approach is the art and the highlighting of the positive side. That it's not just the boogeyman. It's not just a bad thing.
I just think that fear has now gotten into the regulatory arena, particularly in terms of developing drugs, where things are slowed down so much because everybody's insisting on 100%, accuracy with respect to anything we do. And I'm just wondering how that's slowing down, the development of a lot of these processes or.
But personalized medicine, human genomics is pushing it aside. I mean, that'll just flip that on its head. And there's huge progress on that. So.
Yeah, so as a scientist and as a citizen and as a parent, is there anything about the kind of work that bioengineering involves that keeps you awake at night?
That's a good question.
Though if you have young children, you probably just get all the sleep you can.
Yeah, I don't stay awake thinking about work. That's a good question. I don't know. I think I take the busy mom approach, which is like just eat the food.
Yeah.
What's that?
Whatever you eat. The busy mom approach is like, I don't have time to buy them.
But you mentioned state level.
Oh yeah, am I worried about North Korea launching a bioweapons attack? When I think about it, yes. I don't like thinking about it, and I don't think that. I think that's not. Yeah, it's not the most productive thing for me to be thinking about. But people are thinking about it, and people who I think are really smart and are approaching it in interesting ways. So I think there's an interesting kind of like conflict or question, and especially when you're talking about government investment. Like there's a lot of government investment in biodefense, and there should be more, because biodefense it's not just about North Korea, it's not just about the people who are going to do something bad, it's about there are natural pandemics that are coming, it's going to happen, like climate change means more new diseases are emerging, things that we're not prepared for, that's scary. That is scary to me, and that has nothing to do with genetic engineering, but genetic engineering might have something to do with the way that we fight back against even the natural things, right? If you can very quickly sequence a new pathogen and very quickly develop a vaccine and manufacture a vaccine very quickly using a kind of rapid response synthetic biology approach, like that would make a huge impact. That would be a huge benefit. And so there's people thinking about that, and I think that's really powerful. I think anything that you're doing to think about potential like intentional release of a pathogen also talks about the things that are really happening, the new flu that comes every year and kills lots and lots of people. How can we respond to that? How can we be more prepared? I think, so yeah, should we be investing in more biotechnology to keep ahead of the natural issues? I think that's an ongoing kind of discussion, but there's a lot. Oh yeah, I've been looking over here because they've been.
I don't know if this is applicable at all, but I was wondering about the applicability to conservation efforts in working with endangered species. And I was specifically thinking of the Smithsonian's Conservation Biology Institute, which is working very heavily on preserving, I don't know exactly what they do, but working with endangered species, whether your work is, this type of work is informing, the need to collect this material when if they do go extinct or whatever. So if you could just comment on that.
That's a great question. So I think there's two sides, right? I think there is a sense of like, what are we losing? I think you asked what happens when we mess with the microbes, and I think what's really what's the fear of messing with microbes and ecosystems? We don't even know the baseline of what's there. We've already messed with it by messing with the environment, by building cities, by doing all these things. There's so much we don't know about biology and about biodiversity, and so much that will be lost forever, which is just that's a tragedy. I don't even, so people are thinking about it. There's not a lot of money to do that kind of thing. So, and a lot of uncertainty there. There's another side of it too, which is like there's examples of kind of conservation issues, pathogens that are wiping out species. There's a fungi that kills frogs. There's something called like white nose syndrome in bats. There's these things where there could be interventions that people are starting to think about. Where what could you introduce, or how might you intervene using a biotechnological approach to protect those species from extinction? That I think opens up a lot of really, for me, complicated questions like how do you know that it's safe? How are you going to know that you're not introducing something like kudzu or whatever, right? Like there's all those examples from the past of, oh, we'll bring all the rabbits to kill the this, and then the rabbits take over, and then you need to do something else. So like that kind of it's like an Alice in Wonderland, the Red Queen. It's like they send out all the things to kill the things, and this arms race, and the sense of like, anyway, it's complicated, but there's people kind of who are thinking about it, and there's some really interesting thinking happening in conservation circles. There's a gentleman named Ken Redford who's written some really interesting things about how conservationists shouldn't immediately reject biotechnology as part of a concerted solution towards conservation in certain cases. And there's some work happening mostly on islands where people are thinking about what you might be able to introduce within a kind of bounded community in a small island to say, okay, I want to see if I can protect this by doing something over here.
My question was still, last night we talked about how some beans and legumes have things in their roots that store and release the nitrogen.
Yeah.
And driving home last night and appreciating recent rains, we were like, is anybody looking at a way, or whether or not there might be a way to get plants to hang on to water better? And sort of try and, I can't imagine the scale that would be involved, but to try to increase terrarium effect in places that are getting drier and drier.
There's some really nice research on drought tolerance and flood tolerance in different kinds of plants. There's a woman named Pamela Rowland at UC Davis who's done some good research, and she wrote a book called Tomorrow's Table about this. Kind of promoting a different way of thinking about genetic engineering as part of like more large-scale solutions to big environmental problems in agriculture. And she does a lot of work with drought and flood tolerance in crops. And so thinking about engineering those kinds of traits that can be useful.
That was my question. Who is driving the research agenda? I mean, so that if you're these bigger questions that are affecting our society, and Michael's part, you're concerned about the problems. What about the solutions? Who's driving? Okay, we're facing climate change, and so who would then drive that? Would it come from your lab, or would it have to come from the government or a funder, or how? You know what I'm saying? Who determines what's being developed?
A little bit of everything. I'd say it's a little all of the above because of capitalism.
So who comes with a check and drives
money is a big part of it. So the project with Bayer, I think, is a really interesting one because it comes from a pot of money at Bayer that was kind of set aside to say we need to innovate to solve big challenges. Bayer's got a lot of cash, but we don't have a lot of like innovation muscle. What should we do with it? And their approach was to start new companies that they could kind of invest at arm's length and let them kind of like run and be startups, but well-funded startups. And that's where Join came out of. So it started with $100 million out of this fund to have a long runway to approach a big problem that's going to take a lot of time before it gets to commercial reality. And I think right now that's still relatively rare in corporate money structures. People are looking for more short-term, and so that makes it harder to invest in. The government is, in as much as they can, are investing in things that have a longer horizon, but the politics of that and how much that goes in there, that's extremely complicated. But there, in general, I think that there is an idea, there's a sense that the government can absorb some of that risk that companies won't take on, the big companies, and can kind of help seed some of those early ideas. So Ginkgo certainly started with government funding, sort of small grants from places like DARPA and ARPA-E, the energy research projects. And so like long horizon, like not short-term returns, money that was patient, that could help kickstart some innovation. So I think that's the answer.
And that gets into whether the funding is geared towards making a profit center, which is not inherently bad, or whether or not it's geared to just solving
Solving
problems
problems.
that are like, if we don't have a place to live, how much
Yeah.
money we have is sort of irrelevant.
Right.
And
Ideally, you come up with both, the problem solving and the profit generating. So
Yeah.
We were
Well,
at a
if you have like a few $1 billion laying around, like let's talk. Like,
It's like Bill
let's
Gates, you know,
do
that's
it.
global health.
Part of
It's
it
like,
is not only the
is there a
money.
Bill Gates for GMOs?
We did a presentation at Los Alamos not too long ago, and they were talking about their nanotechnology. We got into it quite a bit, and it was absolutely fascinating. The biggest problem is they had no way of really, they didn't have a mechanism to share that knowledge with private industry. And it was sort of like going into a point, there's a lot of good stuff out there, but no one is, it's not money, it's coordination in terms of sharing the information. Because they were talking about all the stuff that they were doing there, and I just asked the question, how are you getting it out? I said, we're having all this trouble, we can't get it out because of all the regulations and all the stuff that's in the way associated with this. And I'm just wondering as to whether or not that's a bigger issue of the work. It's not an economic question or money issue, but more from the point of view of somebody developing highways to get this information out and stop some of the
hard to
competing
make
interests,
something
particularly within the university. They
real.
fight against each other for the same project.
I think there's a lot of good ideas. There's also a lot of work and investment that needs to go into making something real at a scale that is going to make the impact that it could or should. And those questions are often more political and economic than they are technical. And so I think that it is complicated. When you're talking about things that are going to challenge big industries, petroleum, anything, right? Like there's certainly a technical component to it, and tons of really good ideas, and so much energy going into exactly this. Like, what are all the different approaches that we could find? But actually getting up to the scale and to be able to compete with something as big as petroleum, those are economic and political questions largely. And so it's not just technology.
Why don't we end this phase of the discussion?
got it.
I have one more.
Okay.
One more, Michael.
All right.
Just kind of, I think it'll tie everything in nicely. So if you have the resources to work on any project at your lab, you've talked about so many different applications, or maybe you're already starting to work on this, what do you personally find as the most exciting, maybe groundbreaking application that would help better the world?
Beer, obviously.
Beer,
Perception is everybody's
I'm so really into the microbiome. I think there's something coming. There's something there. There's a lot of, it's like a Wild West in terms of
mean
products.
human
microbiome, soil microbiome, all of that. Like
Biomes.
just like rethinking our interaction with the world in terms of like all these microbes that are invisible. I don't know. That's really compelling to me. I'm still into that. I think I have a hard time focusing.
Well, let's thank Christina for We've got lots of food, so stay behind and
Consume it.
Consume.
Your microbiome is great.
My experience in the lab is way different